Now we shall talk about a diagnostic procedure called ELISA. ELISA stands for enzyme linked immunosorbent assay. So ELISA stands for enzyme linked immunosorbent assay. It is a diagnostic procedure mostly used to identify AIDS. Apart from this, it is also used as a procedural method to identify pregnancy, STD disorders, sexually transmitted disorders, thyroid disorders, measles, German measles, which is also called. So ELISA is used to identify all these disorders: AIDS, pregnancy confirmation, sexually transmitted disorders, thyroid disorders, and a viral disorder called German measles. It's invariably used as a diagnostic procedure to identify AIDS, whether the person is HIV positive or HIV negative. This enzyme linked immunosorbent assay with reference to the AIDS is of two types. We call it direct ELISA and indirect ELISA. Direct ELISA is used to identify the antigen, mostly the antigen AIDS, viral antigen, when it is present in the human body. Its existence in the human blood is identified by means of direct ELISA, and indirect ELISA is executed to identify the antibodies that are synthesized against AIDS virus. So direct ELISA is used to identify the antigens that are present in the human blood. In case the AIDS virus enters into the human body, it starts acting as an antigen. So that antigen can be captured by direct ELISA method. And when the AIDS virus enters into the human body, when it starts acting as an antigen against AIDS virus, certain proteins are synthesized, which you call them antibodies. So those antibodies are identified by means of indirect ELISA. Direct ELISA is again of two types. The direct ELISA in human being is of two types. Sandwich ELISA and competitive ELISA. In sandwich ELISA, in case the antigen is present in human blood, that gets sandwiched between two doses of primary antibodies. First, a primary antibody is adsorbed on the microtiter plate. Okay, that primary antibody now sticks to the blood sample containing the antigen. Again, second set of primary antibody is added. So between these two sets of primary antibodies, in case the antigen is present in the blood, that gets sandwiched. Then later on, enzyme linked secondary antibody is added. That secondary antibody goes and sticks to the primary antibody. Then when you add a substrate, that substrate, in case it gives color, it indicates the presence of antigen. If the substrate does not give color by using optical recorder, that is spectrophotometer, it indicates that the person is negative. So that is the main protocol followed in the sandwich ELISA. In case of competitive ELISA, there will be competition between two antibodies, that is primary antibody and secondary antibody. Both of them compete with each other to occupy the epitopes of the antigen. Mostly competitive ELISA is used to confirm the pregnancy. In case the woman is pregnant, okay, the natural human chorionic HCG, the natural HCG present in human urine, and an artificial HCG. Okay, the natural HCG and artificial HCG, both of them can be identified by means of competitive ELISA. Either there is a enzyme linked immunosorbent procedure, it is used to diagnose the AIDS virus in the human body to confirm whether the person is HIV positive or HIV negative. So ELISA, it requires again many chemicals also here now. Okay, like it requires a substrate, it requires an enzyme, it requires even a buffer solution, it requires even a wash fluid, okay, and is broadly divided into two types with regard to the aids, direct ELISA and indirect ELISA. Direct ELISA is used to identify the antigen, indirect ELISA is used to identify the antibody. Direct ELISA, again there are two types, sandwich ELISA and competitive ELISA.